Let me start then by thanking you for the opportunity to come here and present some of our work to you. And I wanted to start out by reminding you then about what you all know, the fact that all cells in our body contain essentially our entire genetic information, stored then in the form of this famous double helical DNA molecule. And as you also know, whenever a cell divides, particularly of interest for us as evolutionary geneticists, of course, is cell divisions in the germline that will then give rise to the next generation. When the cell divides, these two strands come apart, new strands are synthesized, and a sequence of these four bases are replicated in a process that is very, very exact. But nothing is, of course, absolutely perfect. So sometimes errors are made. And if these errors, where a wrong base and nucleotide is built in, is not repaired before the DNA replicates a second time, this will appear as a mutation that may be passed on to the next generation. And we then see the traces of those past mutations in the germline when we compare DNA sequences among humans today. So if you compare a DNA sequence between two people in this room, you will find a difference about every 1,200, 1,400 bases. And because these errors, mutations rain down on the genome approximately as a function of time, if you add a comparison to a chimpanzee, you will find about 10 times more differences, one every 100 bases or so. And we can then use those differences to reconstruct the history of a piece of DNA or a whole genome. In this case, very simply, the two humans go back to common ancestor quite recently, and quite a bit further back is there a common ancestor shared with the chimpanzee. You also know that the genome is composed of about 3 billion base pairs, so there's a lot of information there to reconstruct human history. And if you do that on a worldwide scale, what you will find, and what Alan Wilson found first and on the mitochondrial genome, as we heard, is that most genetic variation among humans is found in Africa. Although, of course, there are much fewer people living in Africa than outside Africa. Everybody outside Africa, if you take them together, have less genetic variation than inside Africa. And not only that, most DNA sequences you will find outside Africa has closely related sequences inside Africa. There is more variation in Africa because there is a component, so to say, of the gene pool in Africa that have no close relatives outside. So the interpretation of this pattern then is that modern humans evolved in Africa, accumulated genetic variation there, and a part of that variation, so to say, went out and colonized the rest of the world. And by using genetic tricks, by seeing how large segments of DNA have similarities outside Africa to inside Africa, we can estimate approximately when modern humans left Africa, and it's less than 100,000 years ago. So quite recently in terms of human evolution. So that is then the genetic evidence for the recent African origin of modern humans. But there is then a problem with this model, if you like, and that is the fact that paleontologists have demonstrated that there were forms of humans living outside Africa since almost 2 million years, other forms of humans, that then these modern humans must have met when they left Africa. Most famously, then Neandertals in Europe and Western Asia, and other forms that are less well understood in East Asia. So our laboratory is then sort of obsessed with studying Neanderthals since 15, 20 years. And just to remind you then, Neanderthals are these robust forms of humans, and they appear about 400,000 years ago or so in the fossil record of Europe and Western Asia. They exist there until approximately 40,000 years ago when modern humans appear on the scene. And you may then ask, why should we be so obsessed with Neanderthals? Why should we be so interested in them? And I think there are at least two reasons to be interested in them. The first one is perhaps the most important one to me, and that is the fact that they are the closest evolutionary relatives to all present-day humans that exist today. So if we want to define ourselves today as a group or as a species, from a genetic and biological point of view, it's the Neanderthals we should compare ourselves to. You may, of course, also be interested in them because they were here rather recently, and you may ask, how are we related to them and what happened when our direct ancestors, modern humans, met Neanderthals? And that's a question that has been debated for decades in paleontology. And there are two extreme views of that. One saying that modern humans come out of Africa, replace Neanderthals with no contribution whatsoever to present-day people. Another extreme view would be that the Neanderthals are the direct ancestors of Europeans today, for example, so almost 100% contribution. And you could imagine anything in between here. And the chance to then begin to address this with genetic means on a genome-wide, comprehensive scale came in the beginning of this millennium, when there came technologies around to sequence millions and billions of DNA sequences efficiently. So you can then extract DNA from a fossil of a Neanderthal, make a library of DNA molecules that you can sequence, you make your database of everything that is in this fossil, all the DNA, and you compare it to the human genome and other genomes and try to make sense of it. And when we then started working on that about 15 years ago, we could already profit then from over 20 years of experience in our lab to work out the techniques to retrieve DNA from fossils without contaminating it with present-day human DNA, as we heard, and manipulating small amounts of damaged DNA in an efficient way. So what you then have to do is you have these sort of short fragments of DNA that you have extracted that's degraded over tens of thousands of years to short pieces. They contain chemical modifications that cause incorrect DNA sequences to be determined, particularly towards the ends of the molecules. So what you then do is map these sequences to the human genome, taking these sort of patterns of errors in them into account, and reconstruct a longer part of the Neanderthal genome. And the first place where this worked was Neanderthal bones from a site in Croatia, in southern Europe. At that time, we worked with 3 different bones and put the data together from them, from 3 different Neanderthal individuals, about 45,000 years old. And in 2010, then we had a first sort of overview over the genome, where we just had a little over half the genome that we'd seen once or several times. And you could begin to ask questions. So one of the questions you were then interested in was this question: what happened when one met Neanderthals, when modern humans met Neanderthals? Did one mix or not? So if modern humans then mixed when they met Neanderthals, say in Europe, a consequence of that would obviously be that Europeans today should share more genetic variants, more alleles with Neanderthals than Africans today would do. Because there have never been Neanderthals in Africa, there's no reason to expect any special relationship between Africans and Neanderthals. So you could then begin to address that question quite simply, because by comparing the genome of a European individual, we happen to choose a French individual as a typical European individual, actually, at the time, and an African individual, finds positions where they differ from each other, and say, how often does a Neanderthal match the European individual or the African individual? Is it 50-50, or is there more matching to one or the other? And indeed, it turned out to be significantly more matching to the European individual. That was surprising to me. Even more surprising was that when we compared a Chinese individual and an African individual, again did we see more matching to the non-African individual. Although most people would think there have never been Neanderthals in China. Papua New Guinea, where there for sure have not been Neanderthals, again more matching. So wherever one looks outside Africa, you find this extra component in the genomes. So that then led to this idea that modern humans, when they came out of Africa, presumably passed by the Middle East, where we know there existed Neanderthals. So if these early modern humans mixed with Neanderthals then, and then became the ancestors of everybody outside Africa today, they could, so to say, carry with them this Neanderthal contribution out to parts of the world also where Neanderthals had never existed. So what has then happened since then is that we have worked a lot with archaeologists in Russia, particularly Anatoly Derevianko and his colleagues, who excavate in southern Siberia, and particularly at a site called Denisova Cave. So it's this beautiful place in the Altai Mountains, close to the border to Mongolia. And they found there in 2010 this toe bones. It's a phalanx of a big toe that turned out to come from a Neanderthal. And by then, from this bone, we managed to then reconstruct a very high-quality Neanderthal genome, of equally high quality as you would determine the sequence, the genome sequence from a person today. So what we can then do with this high-quality genome is we can look at, for one chromosome here, at many people today. Each line would be a person today, and indicate here fragments of DNA that have close similarities to the Neanderthal. And we can then ask, how much do people today carry from the Neanderthal? And that indeed turns out to be 1 to 2% of the genomes of everybody outside Africa come from Neanderthals. But what you will also see here is that different individuals tend to carry different fragments from the Neanderthals. So you can then ask, if we now jump from individual to individual in this room, for example, and try to reconstruct bigger parts of the Neanderthal genome, how much of the Neanderthal genome can we reconstruct without studying any old bones, just looking in people today who walk around on the street? And that's a bit uncertain still, but somewhere 40% or approaching half of the Neanderthal genome is still around today, but distributed among many different individuals, so to say. But as Eric also said in his introduction, this was not the most interesting bones that Anatoly Derevianko and their colleagues found in the Denisova Cave. In 2008, they were very skilled, I think, to find a very tiny little bone and realize that it might come from a human. It's a part of the pinky of the last phalanx of a small finger from a child. And when we sequenced this genome, we were very surprised to find that it was not a Neanderthal, not a present-day human, but something that goes far back to common ancestors shared with Neanderthals. So if we estimate today that the deepest split between populations today, which are between African populations, is about 100,000 years old. Then the split between Neanderthals and the population this individual belonged to is about 4 times deeper, about 400,000 years back. And we debated a lot among ourselves and with our Russian colleagues what to call this group, and ended up calling them Denisovans. So they are called Denisovans after the Denisova Cave, just as Neanderthals are called Neanderthals after the Neanderthal site in Germany. So we can then ask for them, for the Denisovans, have they contributed to present-day people? And indeed they have. You find a small amount of contribution all over Asia from them, less than 1%. And in Oceania then, in Melanesians, for example, it's about 5% of the genome that come from Denisovans. So Denisovans seems to be distant Asian relatives, so to say, of Neanderthals, or sort of the distant relatives of Neanderthals that existed in Asia. So in this picture of things then, between total replacement and total continuity, we have clearly rejected the total replacement idea. So there is this contribution from Neanderthals to about 2% of DNA to present-day people. There's another 5% on top of that in Melanesia. But we should not then forget that a vast majority, more than 90% of all the genetic variation outside Africa does indeed come from Africa. So we often like to call this leaky replacement or so. The big picture is one of replacement when modern humans appear in Europe and Asia, but there is some kind of a genetic leakage a little bit from these other forms. So, and that contribution then seems to have happened early on to modern humans outside Africa. But it may be surprising if one mixed then early in the Middle East, why didn't one mix later on? And I think one did, and we begin to see evidence of that. So for example, in 2010, there were cavers in Romania, in this cave, who found a mandible of a modern human that turned out to be very old. It's about 40,000 years old. So this modern human lived in Europe at a time when Neanderthals were still around. So we were very interested in studying that genome to see had one already mixed with Neanderthals or not. And indeed one has. So this is one chromosome again, indicated in color from some present-day humans, fragments that come from Neanderthals. And if we now look at this 40,000-year-old individual, you find a lot more Neanderthal DNA. And indeed, these segments that are black here, we just have no information because this is not a complete genome we're looking at. So it turns out that for this chromosome, more than half the chromosome is solidly of Neanderthal ancestry. And you can find 7 such fragments around the genome in this individual. So this, of course, indicates that this person had a close relative who was Neanderthal. And you can model that and show that it was 6, 5, or 4 generations back was there a Neanderthal ancestor here. So we sort of almost caught someone in the act, if you like, of mixing with Neanderthals. So we then know that there is sort of this extra mixing going on later on here. And I think that may have happened many times. And I think we will be more in a position to study that in the future, because what has happened in the last year is that we have started to study DNA retrieved just from sediments at archaeological sites in caves. So instead of studying bones, studying just archaeological sediments like this. And indeed, you can retrieve DNA from such sediments. So these are 8 different sites we've studied, 3 of them in France here, where you then find DNA from animals, so woolly rhino, hyenas, cave bears, and so on, and sometimes also of humans that have lived in those caves in the past. So you can then look through the stratigraphy, for example, in Denisova Cave, and not only determine where humans have been thanks to bones you find, and DNA in bones, but also just sediments. And you begin to see a complex picture, where in this case, deepest down, you find Denisovan DNA, and Neanderthal DNA, and Denisovan again, Neanderthals, and Denisovans. So I think in the future, I hope we will be able to reconstruct sort of the population sequence in caves with much greater detail like this. So for the last few minutes then, I wanted to change gears a little bit and sort of say, they have this contribution from Neanderthals and Denisovans to present-day people. Does it mean anything functionally for people who live today? And we're beginning to learn quite a bit more about that. So just to give you an example, for example, in Tibet, there are genetic adaptations for living at high altitude where there is little oxygen in the air. That, for example, means you have much less problems in pregnancy if you have these genetic adaptations than if you're, for example, a Chinese person who has moved to Tibet. And it was also already known that one of the major adaptations is a variant in this gene called EPAS1 that has to do with how much oxygen you can absorb without changing the number of red blood cells in your blood. It's very frequent in Tibetans, like 80%, hardly seen anywhere else. And Rasmus Nielsen's group at Berkeley, with the help of the Denisovan genome, then showed that this variant has come over from Denisovans into the ancestors of Tibetans. So probably you would not have so big populations living at the Tibetan high plateau today if there wasn't this variant coming over from Denisovans. Some things seem to have been so important that they have come over both from Neanderthals and Denisovans to present-day people. So this is one such example, a cluster of toll-like receptors. So these are cell surface receptors that are important in innate immunity. They occur, these variants, outside Africa. And you can, if you reconstruct their evolutionary history, you can see that some variants have come over from Neanderthals, other variants of these genes from Denisovans. You can then go on to show that people who are homozygous carry two variants of these Neanderthal variants here, express more of these receptors than people who express just one variant or no variants. And you can show that carrying these archaic variants is associated with increased resistance to Helicobacter pylori infection, so the type of bacteria that give you ulcers, and also an increased tendency for allergic problems. So you may perhaps blame the Neanderthals if you have allergic problems, but thank them if you don't have an ulcer. So Josh Akey's group have recently made a review of things that seems to have been positively selected in human ancestors and come from Neanderthals. Many of these variants have to do with immunity and also with skin and pigmentation. But we should not forget, so there's sort of a idea coming that these modern humans come out of Africa, meet these other groups that lived for hundreds of thousands of years in Eurasia, have genetic adaptations to the environment there. They pick up some such variants that then help modern humans to get along better. So it's something we would call adaptive introgression or so. But we should not forget that most variants today seems to be negative. So most Neanderthal variants you find in the population and find an association with them are associated with diseases, things like hypercoagulation, many skin diseases, even things like depression, and so on. So there seems to have been negative selection overall against most of the variants coming from Neanderthals. And you can also see that if we go back in time. So today, as we have said, 1 or 2% of the genomes of Europeans come from Neanderthals. If we go back to older genomes that many other labs have produced that are a few thousand years old and older, you will see that as you go back in time, you find more and more Neanderthal DNA. So the best explanation for this is sort of selection against it, pushing it out of the population. And before ending then, I wanted to bring up one more thing, and that is sort of the question, what was not contributed by Neanderthals to present-day people, if you like, or by Denisovans. So you can now go over the whole genome, chromosome by chromosome here, and mark in red the frequency with which you have contributions from Neanderthals in Europe, and in green where you have it in Asia. And you can ask, are there parts where you would expect a contribution but you don't see it? And indeed, there are such areas. And they are very interesting because they might hide things there, variants that are really unique for modern humans, so we select against Neanderthal contribution. So things that changed here after we separated from Neanderthals. And they are so interesting to us because they may be behind this fact that modern humans technology starts changing very rapidly. They start becoming figurative art. Modern humans start spreading across the whole world. And we're now in a very interesting position, since we can identify all these changes and make a catalog of them. Changes in all present-day humans where Neanderthals look like the apes. And that is not a very long list. It's just around 30,000 changes. And I see that my time is almost over, but just to give you a little feeling for how we now begin to address this thing over the next years, we can just think about the amino acid changes in proteins. There are just 96 such amino acid changes in these genes here, and we of course want to ask which are important. So we have our bias. It should be things involved in brain function or so, because we think modern human cognition is special. And if we now look just in different parts of the developing human brain, we find particularly many of these proteins expressed in the proliferative zone where stem cells divide. So, and that is due to that there are 3 proteins there that all have to do with the metaphase plate, with the spindle and kinetochores when chromosomes are pulled apart during cell division. So far we just speculated, saying this might be due to that when the stem cells divide, it's very important how they divide in terms of how many neurons are formed. But we can now begin to ask this directly with the help of stem cells and iPS cells that you can differentiate in vitro to cerebral organoids, these little models of developing brains. So we can now make such models for human mini brains that develop in tissue culture and chimpanzee brains that develop in culture. So when we do that and compare them in our lab, they look very similar to each other. But there are some interesting differences. And this is work by Maura Bermudez that collaborate with us in Dresden, Wieland Huttner's group. So when you now look at the cell divisions of these apical progenitors and look at the cell division here, when you see the chromosomes line up here in a plate and then are pulled apart in this metaphase, you find that this period of the cell division is much longer in humans than in chimpanzees. And that is specific for these types of progenitors. We don't find it in other cells. So that makes you think again, of course, about these 3 proteins here with these changes. So what we are now in the process of doing is, of course, to engineer in these changes into human stem cells to make these brain organoids and also put them into the mouse to study brain development, to see if that is involved in some aspect of brain development, these changes. So to end then, I hope I convinced you that if you're interested in human evolution, it's very valuable to have the Neanderthal genome. You can find out things about the history back here, but you can also then focus in on genetic changes that are unique to modern humans and not shared with Neanderthals and other primates. And the way in the next few years to study that will be to put these genetic changes into stem cells and study their effects, and into humanized mice. And I should of course mention that many, many people have been involved in this, many paleontologists that I will not have time to mention. I mentioned one person in the lab, Matthias Meyer, who developed methods that allowed us to make these high coverage genomes that are so important also for the functional work. And many people have analyzed the population genetics aspect, particularly Janet Kelso, who coordinate all the bioinformatics in our lab, and David Reich and Monte Slatkin for developing population genetic methods to understand these genomes. The functional work involves particularly then Wieland Huttner's group in Dresden and Barbara Treutlen and her group in Leipzig. And with that, I then thank you for your attention.
Thank you for this very nice presentation. We can have just a few questions. Donc, Moshe Yaniv.
Thank you for this very beautiful and instructive talk. Just a question. The Neandertal and Denisova went out from Africa also, and when? The same time or different times?
So So most likely their common ancestor is something paleontologists would call Homo heidelbergensis, or so, that you find fossils in Africa and then also outside Africa. So probably they left Africa in the order of 500,000 years ago or a bit more, and then evolved in Asia to Denisovans and in Europe to Neanderthals, would be the idea. So it would be an earlier out of Africa event.
And they never went back? They never went back to Africa?
No, as far as I can tell, they never went back. No one has found anything with a Neanderthal morphology inside Africa. So it's really a European, Western Asian thing. And the morphology of Neanderthals also become more and more extreme as time goes on towards the end of their history.
Pierre Corvol, just in the back here.
Well, thank you very much. If you look at the Neanderthal genome, and if you look at genes involved in reproduction, FSH, LH, and other genes as well, is there any variations which could account for, let's say, a less reproductive species than the humans?
Yes, so there is some hint that there might have been a problem with reproduction. So if we just look for genes expressed in different tissues and ask the question, is there an underrepresentation of a Neanderthal contribution in any such groups of genes? There is one thing that stands out, and that is testis, so the male germline. So there is some hint that there might have been problems in male hybrids, which would sort of fit with other types of reproductive problems between closely related populations or species. That's the only sort of hint that there might be some issue there. Well.
Daniel Louvard.
Yeah, okay. Beautiful indeed. You showed a curve of the fact that the Neanderthal genome is gradually reduced over time. Can you extrapolate when there will be no contribution, or do you think there will be a limit?
Yes, I guess we could. We have not extrapolated into the future.
But there may be a reason not to eliminate.
Yes. I mean, there are clearly some things that are positively selected. There are things that have risen to 50-60% allele frequency, and they will probably always stay. I would also believe there are lots of things that are neutral and woods. And I'm a little always scared to show that decrease too. It's sort of extrapolated after all this decrease from rather few points, as you see there. So, yes.
Jean Weissenbach. This way.
On the same line, would this explain that you don't find any remains of Neanderthal in Africa?
So saying that it would have been selected away a contribution in Africa?
No, just this decay.
Yeah.
And since, well, they moved, maybe if they moved, also some others remained in Africa.
Mm-hmm.
So how do you explain that you don't find any admixture of Neanderthal in Africa?
Well, I think that sort of the ancestors of Neanderthals then left Africa sort of somewhere over 500,000 years ago, then was more or less separated from African populations, which included then the ancestors of modern humans, which then later on come out and meet them. And that's in a sense why we can detect this mixture, because they were separated for quite a while, so accumulated differences, and then we can detect when they come together. There is now a little bit of evidence that there was genetic contact, also earlier between modern humans and Neanderthals. There is some evidence that there was some gene flow from Africa to Neanderthals more over 100,000, maybe 200,000 years ago. But there has not been a lot of gene flow between those continents.
Last question by Pascal Cossard.
Okay, so the Neanderthal. Before them, you had the monkey. So I would like to know, to have your comments about our genes coming from the monkey through the Neanderthals to us. I mean, can you tell us a little bit more? I'm lost.
So,
How many monkey genes I have?
well, of course, we share a lot ancestrally with other groups. So we have a common ancestor with chimpanzees somewhere 13 million years ago. But then there is no gene flow with chimpanzees anymore. Then we have a common ancestor with Neanderthals, say 600,000 years ago. So we share a lot of variants just because of common ancestry. But there is this later trickle over from Neanderthals that bring over these later variants.
We have to discuss.
We have to discuss later, yes.
Still another question down there. Yes.
Yeah.
Is there enough information, sequence information at this stage, from enough layers to be able to detect any bottlenecks that would coincide with climactic or other environmental events that might have led to a near mass extinction and then expansion of some specific genotypes?
Yes, so I think we have actually recently determined a second European Neanderthal genome to high quality, and we have a number of genomes to sort of 1-2-fold coverage. So one begins to be able to reconstruct sort of the genetic history of Neanderthals. And then we will see what happened during the penultimate glaciation, for example, or glaciation before that. And the hints that we sort of see in the data is actually that there were other Neanderthals in Europe that disappear in the second to last glaciation. And then there might be a recolonization actually from the Middle East and even from the Altai Mountains. Because we see that this Altai Neanderthal there is closer related to late Neanderthals in Europe than to early Neanderthals before the second to the last glaciation. So indeed, we will begin to understand some of their history.
All right. Thank you very much again.